The popularity of obesity in the world is increasing at an alarming rate. An article published in 2011 suggests there may be an answer for those who have an irregularity in energy homeostasis, resulting in these high fat levels. The paper, written by George Lameau and his team of scientists, is aimed at discovering how certain compounds affect fat storage in Caenorhabditis elegans, a nematode which proves effective in whole organism screening due to its transparency. 3,200 compounds similar to common pharmaceuticals were tested on the C. elegans using Nile-Red screening. 1% lowered fat levels by over 2 times the control, 0.4% elevated by the same amount, and 1.2% caused lethality or poor health. 10 of these compounds showed high impacting phenotypes and were further investigated. F21, F17, and A13 all showed dose-dependent reducing effects. The team then observed the effects of the expression of FAT7, which regulates the conversion of unsaturated to saturated fats within an organism. Nematodes expressing FAT7 GFP, which is a green fluorescence, were tested with 10 microns of each compound. These results clearly parallel that of the Nile-Red screening earlier. One important thing to note is that growth, feeding, and the ability to reproduce were undeterred by all of the compounds. The next study was conducted to find if these compounds showed effectiveness in mammals through mice cells. 3T3L1 cells had shown an abundance of lipid droplets prior to testing. A13, I13, and F21 all showed dose-dependent reduction. However, A15 showed an increase in volume and number of lipid droplets. Because only 4 compounds showed activity in the mice cells, it can be said that the remaining 6 were C. elegans specific. HepG2 cells in the human liver were analysed to observe F17's effects on AMPK function in humans. Lipid-specific staining revealed numerous lipid droplets in the cytoplasm of each cell. Treatment with F17 significantly reduced this lipid staining. Several more compounds were then tested. ACAR and F17 induced phosphorylation of acetyl-CoA, however F21 did not. This shows that F17 is a compound effective in metabolism regulation in both humans and nematodes. With further research into these compounds, it's likely we'll see more effective pharmaceuticals in the near future. Although these authors haven't released more on the topic, 2 scientists, Chow and Sato, have recently worked on fat regulation in C. elegans also. Although they've used different compounds, they've maintained similar results through similar methods.